And with that, I will hand things over to Tanya. Thank you.
Thanks, Dan, and welcome everyone, and thank you for spending part of your evening with us tonight. I'm Tanya Isa. I'm a member of the board with Hudson Taconic Lands, and as always, we will begin with our land acknowledgement.
What is this?
It's on the fungi.
Oh, and please mute yourselves through the duration. Thank you. It is with gratitude and humility that we acknowledge that we are learning, speaking, and gathering on the ancestral homelands of the Mohican people, who are the indigenous peoples of this land. Despite tremendous hardship in being forced from here, today their community resides in Wisconsin and is known as the Stockbridge-Munsee community. We pay honor and respect to their ancestors, past and present, as we commit to building a more inclusive and equitable space for all. Next slide, please. Thank you. This map is a testament to what tremendous community support makes possible. Hudson Taconic Lands has conserved over 18,000 acres in Rensselaer County and stewards more than 20 public nature areas. The darker green shows our conserved lands. Lighter green marks other parks and preserves around the county. And the red, that's our most recent work. Those are lands protected in just the last 5 years, many of them made possible through our Forests Forever campaign. Next slide, please. And now I'm delighted to introduce tonight's speakers. Dr. Patricia Kaishian is the Curator of Mycology at the New York State Museum, where she oversees one of the region's most significant fungal collections. She is also the author of Forest Euphoria, The Abounding Queerness of Nature, which has received wide acclaim, including recognition as one of Time Magazine's 100 must-read books of 2025 and a Vanity Fair Best Book of 2025. Dr. James Lendemer is the Curator of Botany at the New York State Museum, where his research focuses on lichens, the remarkable symbiotic partnerships between fungi and algae that colonize ecosystems from Arctic tundra to city sidewalks. One of North America's leading lichenologists, he has described hundreds of new species and is a leading voice in biodiversity documentation and conservation. So welcome to our speakers. We're thrilled you're here. We're thrilled all of you are here. And just a really quick reminder that if you have any questions, please put them in the chat, and James and Patty will answer as many questions as time allows at the end that I will pose to them from the chat. So please stay on mute. Thank you.
Hi, everyone. Can people hear me?
Yeah.
Okay, great. Yeah, so I'm really happy to be here. I've been conducting fungal biodiversity surveys on land within the Hudson Taconic Lands now for several years. And my goal here tonight is sort of to walk you through some common and beautiful or just interesting fungi that I've seen, mostly at Valentino Family Community Forest, which is where I do a lot of my surveys, and sort of give you an introduction to basic fungal diversity survey methodology and just sort of kind of chat and explore some of the amazing things that I see in the forest. As Tanya mentioned, I'm the curator of mycology at the New York State Museum, and it is a historical collection. It was founded by Charles Peck, who was a botanist, but who was very interested in fungi in the early 1800s. And he dedicated his many years at the museum to documenting fungi throughout the state. And he was one of the first North American mycologists, and he's worked very prolifically to describe thousands of different species And many of the fungi that you might commonly see in the forests of this region are specimen or are species that Peck either described himself or collected or worked on in some way. This area generally was his home, and he did a tremendous amount of field work here. So it's sort of cool as a scientist and mycologist to be able to visit a lot of the areas that Peck worked and look for the same fungi and then add them to the collection a couple of hundred years later. I am the first curator of mycology since 2005. So I started at the museum in 2023. My predecessor retired in 2005. So there was this 18-year gap where there wasn't really much mycological research happening. So when I took this position, I was really excited to not only be doing like steward work on the collection, but also be doing field work. And the way I found out about Valentino Family Community Forest is I simply Googled a public land that was close to my house when I moved to Troy in 2023. And it caught my eye because it had just recently been open to the public in that summer. So I was really excited to go to a place that maybe didn't have a ton of foot traffic yet and was sort of close by, and just I don't know, there was something about the pictures that really enticed me, and I had this hunch that it would be a great place for fungi. So I visited it for the first time in June of 2023, and I was completely correct. I was spot on. It was an amazing place for fungal biodiversity. The It's a very like interesting dynamic habitat, lots of different tree stands. There's lots of hemlock, white pine, beech, maple, hickory, oak. And it's just topographically very variable. It seems to get a good amount of moisture. And so I was just really dazzled by the amount of fungal biodiversity I saw there that summer. And I decided to start a biodiversity inventory of that area, which my goal is to make basically a long-term survey over the course of my career at the museum. So I started this in 2023, and I've made more or less weekly visits to that forest since then, with the exceptions of like the dead of winter. I don't tend to go November. I tend to not go like December through February, and then I kind of tepidly might go out in March, but usually just as an excuse to be out of the office, and I don't find too much yet at that time of year. But most of the time I go spend a whole day there every week documenting fungi. So fungal biodiversity is really immense. There are millions of species of fungi estimated to exist, and we've only described maybe 3 to 5% of them. And New York is actually, this part of the state is home to a really good amount of fungal biodiversity. We get a ton of rain in this region, and the like humidity and sort of seasonal variation that we have supports a great amount of fungal biodiversity. So to catalog all of these things actually takes a lot of time. Also, fungi are really ephemeral in many cases. Of course, you do have some species that are persistent all year round. But for the most part, when you're thinking about macrofungi, they produce these fruiting bodies that are temporary, and you don't necessarily know exactly when or where they will arrive or emerge, and you don't quite necessarily know when you will see them again. So long-term biodiversity surveys are really important to really understand the character of that population, like what species exist here, what are their population dynamics like, and to do that, you really need to be visiting a place again and again and again for years to really understand the extent of the biodiversity. So I just want to walk you through. I'm going to like chat about some of the species that I have up on the screen, and then kind of give you some other little anecdotes and information about my surveys. So this is Cantharellus flavus or flavus. This is something that many of you might recognize as the chanterelle. I chose this first because it's pretty iconic to the region, and people know it as a culinary mushroom. It is delicious, in fact. And these I have found in this area. They're pretty common in the summer here in the capital region. And this one you can see was quite large, almost like 6 centimeter or 9 centimeters tall, 10 centimeters tall even. So really beautiful bright golden mushrooms. So what I do is I'm my first few years were spent collecting mostly everything that I was seeing. This is Suillus americanus. This is another really common, this mushroom I see almost like all year, like all of the mushroom season, which is peak in June, July, August, and early September. I would say the real peak of mushroom season in this part of the world is July, August, and early September. But I'll find this mushroom pretty much out all summer into the fall. Um, and so I'm really interested in the first couple of years. I wanted to just start a species list. I wanted to say, okay, these are the species that I'm finding, and then I wanted to make really nice collections of these specimens to be added to the herbarium. So I went about this in what we call an opportunistic sampling method. Basically, that means I'm just walking around looking for anything. I'm not walking in a particular line or covering a fixed area. I'm strolling and ambling around using sort of my experience as someone who's combed the woods for mushrooms for many years, just to figure out where are the mushrooms growing, where are they most abundant, and where can I find them. And then I'm using, these are all photos I've taken just with my cell phone in the field. I always take cell phone photos. I sometimes go out with a nicer camera when I have more time, but in the early phases of this project, I'm like pretty busy most of the time I'm out in the field because there's so much ground to cover and specimens to collect. So I hope down the line I'll take more like publication grade images of my findings. But I'm always taking fresh photographs of mushrooms because when you collect them, they in their dehydration and sort of in their movement, like the movement they're subjected to, their characters can change and it can make them harder to identify. So a nice photograph will kind of show the mushroom in situ and it will show key characteristics at usually like many different angles. So I take photographs from above, I take them from below, I take them from the side. I arrange the mushrooms in like an array so you can see the growth across like the maturation process. Mushrooms can change really dramatically from new specimens to mature specimens. So I like to show even in just one photograph sometimes that sort of arc of maturation. So in going through the forest, I'm looking for anything that I can just put on my list. Sometimes I can identify it right on site in the field, but because there's just hundreds and hundreds and hundreds of species of macrofungi in the forest, oftentimes I don't know what it is just off the top of my head, and I have to take it back to the lab to identify it. That involves sometimes looking at the spores under the microscope, sometimes using dichotomous keys to sort out all of the features. And that process can be really time consuming. It can take me sometimes several days to identify a mushroom or fungus. This is an example of one that's very easy to identify. Actually, the ending that got a little cut off on the side, but it's Clavaria zolingerii. And this one is really just unique looking. It's this beautiful, vibrant purple coral, and there's really no confusing it. So this is an example of one that is pretty easy to and classic for the region. It's common in that I see it most years, but it's usually not found in a tremendous amount of abundance. And that's the other thing I'm interested in. I'm interested in understanding how often, so I don't want to just collect a mushroom and put the species list, put it on a species list. I'm also interested in knowing like how many weeks in a row am I finding it? And I take notes, really detailed notes about other aspects of this encounter. So am I finding it in, high numbers? Are there large patches of it? Is it like a really robust growth? Or was it this really straggly, sort of strange one-off encounter that I would never have expected and might not encounter again? This is something called Hygrophorus eburneus. It's a really beautiful mushroom, kind of waxy like mushroom. It has a slick, a viscous top. And one thing about mushroom ID is that there are so many features you have to pay attention to. It's rarely just enough to look at a photograph, especially for a beginner, and know what something is. Sometimes things can look at first glance really similar, but what you have to do is actually triangulate a bunch of different features to confidently ID it in most cases. Again, if you see that purple coral, you're like, okay, boom, that's what it is. But if you're trying to identify like a small white mushroom, you're going to have to look at how the gills attach to the stem, the surface texture on the cap, the overall coloration, the proportion of like the length of the stem to the width of the cap, whether it's growing in soil or like here in the pine duff amongst some moss. You can see there's a maple leaf here, so maybe that's going to tell you something about the habitat and maybe like the ecological role of the fungus. And all of these things are so important to be able to identify it. Also not pictured here is the spores. Spores are really helpful identifiers in fungal identification. So the color, the shape, the size, the ornamentation on the surface of the spore, all of that is like diagnostic, and we are just as mycologists very often looking at that. This is another one that people are probably a bit familiar with. This is Trametes versicolor, or the turkey tail. It's really beautiful. It's a wood decay fungus. It looks like it's just growing out of moss, but it's a moss-covered log. So knowing the substrate again is really important in fungal identification. So because it's growing on wood, and we know it's wood decaying, meaning it's breaking down the lignin and cellulose in the wood and releasing the bound up nutrients and participating in the like sort of nutrient cycle. This is something that grows seasonally, but like the remnant of the tissue can be remnant for many years. So like it has a growth period, but in the fall, but then it is a leathery sort of corky texture that is persistent, so that you can often find this like throughout a lot of the year. But often it's not, it's past its, if you find this in the dead of winter, it's not actively producing spores. They would have already been disseminated. This is another really common mushroom we find around the forest. So the genus is Russula. This is Russula america, but I will say this genus is really difficult to identify things to species. They have there's a lot of red-capped Russulas that look really similar. And this one, the name emetica means like an emetic, meaning something that would make you actually vomit. And the common name is sometimes the sickener. And what's helpful is that if you take a little nibble of it, it tastes horrible, so you won't be enticed to eat it. Amazingly, people are really afraid of mushrooms. Obviously that's changed a little bit recently in the last 5 or so years. There's been a huge uptick in interest and intrigue within mycology or of mycology. But, a lot of people still ask me, like, is it safe to handle mushrooms? Can you touch them? Like, what kind of precautions should I take when I'm interacting with them? And really, it's quite safe to handle all mushrooms that you would find in this area. There's nothing that would happen to you from simply handling the mushroom. I have met literally one person in my life who gets minor skin irritation when they handle some mushrooms, but seems to be sort of like a very idiosyncratic like allergy that they have. All mushrooms are safe to handle. There are some extremely poisonous ones that we would find in our forests around here. This is not extremely poisonous. It just would make you it would just give you an upset stomach. I mentioned the taste because we actually, as mycologists, do taste test mushrooms to help us identify them sometimes. But the next part I'm about to say is super important, which is that we always spit out the tiny nibble we take, we always spit it out. So one way we actually identify Russula species that are the same color is taking a tiny nibble and seeing if it's really acrid, really spicy, and kind of like unpleasant, or if it's very mild and neutral. And that is like a very common step in identifying this group of mushrooms, which is a little fun. These are like, this is something called Geronima subclavatum. I'm using a lot of the scientific names because, to be honest, a lot of these do not have broadly established common names. I'll mention the common names when they exist and they are in regular use, but there's so many species, and mushrooms are so poorly studied that there hasn't been that sort of effort to standardize names like the way we've done with birds or trees and things like this. So I'm not doing it to be, any like elitist or try to make people feel intimidated. The opposite, I would love for more people to study mushrooms and get excited about them. But this, for example, doesn't have a common name. I want to point that this mushroom, these photos I included because one kind of cool characteristic is that sometimes you can see spore deposits on the surface, the cap surface of a mushroom if they're growing close together. If one cap is sort of beneath the gills of another, like those spores might drop onto the cap, and you can see then in the field what color what color spores you have with that fungus. So on the large photo it's my right, I'm not sure if it's mirrored, but it's the one that doesn't have two on top of each other, like the top down view of the mushroom cap. You can see that dusty sort of orangey brown buff color, like what we call, that's a mycologist pink actually. Um, and that's a spore print from a neighboring mushroom. And spore prints are just so helpful because of how significant they are in like the phylogenetic tree, like basically the phylogeny of the mushroom. So color is, sometimes you even find guidebooks that are organized by spore color. So knowing the spore color right away just helps you identify it. Also, this I arranged for the photograph, so I, help for like, so I could remember this like better later. I arranged them on this highly decayed log that they were growing on, which helped me identify them as well. This is another really beautiful mushroom, grows in the fall here. It's called Ichnoderma resinosum. I think sometimes people call it like the resinous polypore. Um, and it gets its name, as you can probably tell, from this exudate that's forming coming from the fruiting body. And it's almost always there on it. I don't quite know why this fungus produces so much of it. Like not all of mushrooms do, but it's a it's a part of its metabolism. It's just sort of a metabolic byproduct. So it's not known to be, at least as far as I understand, it's not known to be particularly like functional. It's just like a byproduct. This one is a polypore. This morphology, it's what we call this type of like body plan. And it grows like polypores are like in the classic sense, grow like shelves out of wood and have underneath their cap like many pores, so polypore. So instead of gills like you would see in this mushroom, there are pores underneath, and that's where the spores are ejected from. This one, I love the texture of it. The white tissue around the margin is the new growth, and it's very soft and pillowy, and I just find it like as a sensory thing, like very, very pleasing. These two I just thought looked really amazing together because of how different they are. I'm sorry that one looks cut off too, but so Clavulinopsis helvola, that this is like a type of like a club, kind of in the coral general body plan category. And then this is a Cortinarius albovilaceus. And I just think this is a striking photo to me because these are not like, they're distantly related, but they're in the same phylum. They're not like, there's two major phyla in macrofungi that we encounter regularly. And these are in this, you would think they were completely on two different ends of the earth within the kingdom, but they're not. They're in the same phylum, Basidiomycota. So it just gives you like this sense of like how diverse morphologically fungi really can be. This is another really iconic mushroom in the Hudson-Taconic Lands area. This is Ganoderma sugi. The epithet sugi comes from suga, the hemlock, because it exclusively grows on hemlock. As I mentioned, there's a lot of hemlock at the study site that I visit, and so I find this really consistently, mostly in July is when it's like freshest and there's the most abundance. So I'm always cataloging, like each week, if I'm finding something, I collect it, the first time and maybe a couple times to get a few really high quality specimens for the museum. But then I'm not interested in just taking, right? I'm very concerned in general with impact and like sort of conservation as well. So the idea is to get a good representative specimen that you can use scientifically for, all sorts of things really, but that vouchers this record. But then I catalog occurrences just by documenting it. I always photograph everything. Some people say, you know, without a photograph, like it's just or some without a voucher, it's just a rumor. And then, but next best thing is a photograph and really detailed notes. So I so like again, throughout the summer I'm seeing these Ganoderma, the hemlock reishi is the common name. So I'm seeing these hemlock reishi like week after week, and I'm not collecting them every time. And in part because they're habitat for other organisms. I don't want to interrupt the fungus's ability to reproduce. And you know, in general, just sort of like a leave no trace mindset. A lot of people ask me about like the impact of collecting fungi, and there is this idea that it's not like, you know, collecting plants because you're just taking the fruit. An accurate and like for a lot of mushrooms, it's an accurate analogy to say that the mushroom is like the apple on an apple tree. So taking an apple is not the same thing as uprooting the whole tree. And that's true for a lot of different mushrooms that you'll commonly encounter. But again, they're also habitat. They're for invertebrates. They're a food source for mammals and invertebrates, and reptiles and amphibians. And we don't always know if the fungus is mature or not, or if it's reproduced all of its spores, and if there's some other stressor that might be involved in taking it. How does that sort of affect that individual fungus? So a lot of these things are unknown or, you know, not clearly documented. So I do try to be mindful about that whole process. This is another really common thing you'll see. This is Listeria austria, or also known as false turkey tail. They're similar to the turkey tail in their growth habit, and they have these like a similar concentric banding on the top, but their underside is really smooth, and the pores, this is also a type of polypore, are basically invisible, and you can see them with a hand lens. Just checking the time. I'm at a couple more minutes. This is a beautiful mushroom, Romeriopsis kunze, or people call it like a white coral. I'm not sure if it has a really like a widely adopted common name. This is something I see a lot in the late summer in these areas. Just really beautiful. This is another really common, I forgot to put the name in the slide, but this is these are hygroscipes, or the wax caps. This is a kind of you know, like there's a handful of species in this genus that you can find in the area. They love boggy, mossy areas. They grow from the soil, meaning they're saprotrophs. So they're breaking down dead or dying organic matter. So like wood decay fungi are also saprotrophs, and that's as opposed to being something like that's mycorrhizal, which is forming those mutually beneficial relationships with plants. These are we call them this like the ketchup and mustard group, because a lot of the members of this genus are somewhere in the color palette of ketchup and mustard. I think they're so cute. This is another really common mushroom. This is one of the first large macrofungi you'll find in the late spring in this area. It's called Megacolybia rodmanii, and also known as the platterfull mushroom. They can be enormous. This is the biggest one I had ever seen, and this was like like one of the first trips I had made to this forest during my field work, and I was just really stunned by by the size of this one. This is another, you don't see the whole fungus, but I wanted to more capture like the fact that fungi are sort of these little worlds unto themselves, hosting a number of different species, doing all sorts of different things. So you have like a little slug, and then you have these beetles that they lay their eggs in the tissues of this fungus. So this is also known as the artist's conch. Maybe some of you have seen these elaborate, beautiful drawings that people can kind of press into or like basically by just using pressure, they can stain the underside of this fungus. And then if you dehydrate it, that whatever you've drawn onto the surface of the underside will remain like sort of fixed for decades. And the little beetles here will like burrow into this tissue. It's very hard, very woody, and it's a great safe place to raise your kids. So that's what the beetles do. I one time collected you can also find these in the other Ganoderma, the hemlock reishi. One time I collected some hemlock reishi and I brought them into my bedroom to dry in the window. And for the next few weeks, I kept finding these beetles on my pillow because they kept emerging from the fungus. Um, some more, another nice capture that shows like just the diversity of form. The one on the right, or the mushroom looking one, is a bolete. That's the type of like sort of body plan. So instead of gills, you also have pores, but instead of like the polypore, which is like flat and shelf-like, usually this has a mushroom kind of body, like the physique of a mushroom, I guess you could say. The common name for this one is the bitter bolete, and similar to that Russia America, you can immediately tell if you just poke it with your tongue. Like on the other flip side, the Clavulina cristata is this little coral mushroom, and those are actually edible. On that, but I just, to do my due diligence, never eat a mushroom that has not been identified by an expert until you are yourself one. And do not use AI to identify things if you're going to eat them, or for really any purpose, but especially if you're going to eat them. It's not good enough yet. Okay, I'm going to sort of cruise through these last ones just to show you them, and then we can move on to James, and then I'm happy to just talk about whatever comes up for you in broad mycological questions, specific mycological questions, or whatever you're inspired to ask. Sort of a whirlwind tour, but there's so much to cover. So I will quickly say, this fungus, I've never seen it in such abundance at any other place, but in this area. These are so cool. These are jelly tongues, Pseudohydnum gelatinosum. This is the underside. They have like teeth. That's what we call those structures. This is how I arrange my data, like my species list, the dates I found them. And these are some of the data when I take my notes, I'm filling out all these sections very intensively. Okay, that is it for me. So I think if someone else can exit my screen share. I'm not sure. Can people still see my screen?
No, your
Okay.
screen is
Okay.
gone.
Okay, perfect. Okay, thank you.
Thanks.
But now, can you see my screen?
Not yet.
Oh, really?
It says that you have now we can see it.
Great. Okay. I wonder why there was that lag.
It's not in slideshow mode just yet. Might be a lag.
Yep.
There we go. Thank
Okay.
you.
If there is a lag between this and the next slide, just let me know, and I can turn my camera. Actually, it might help to turn my camera down. Wi-Fi should be okay. Anyway, so thanks so much, Patty. I cannot help but just be awed by the diversity of fungi that are out there. And even though I study fungi, the lichen world is like a little bit different. So it's just kind of funny because we both work on fungi, but there's so many species and there's so much diversity in size, shape, form, everything, that it's just like it's almost like being on opposite ends of the earth in some ways, even though these things are relatively closely related to each other. And so, what I wanted to do was to take the opportunity to talk a little bit about lichens in general, sort of what they are, and how our local lichens fit within the broader lens of sort of the big picture of Eastern North America and Appalachian or Eastern temperate deciduous forests. And then also some of the conservation take homes from work that my colleagues and I have done over the last decade or so. And then hopefully convince you all that what we really need to do is make more of an effort to understand lichens everywhere, including here in our home in New York State. So lichen diversity is fungal diversity. Let's start with fungi. There's millions and millions of species of fungi. And some of these are the same species that Patty showed. They're all so photogenic. They're so amazing. And I just think, again, it helps to just take a step back and say, like, wow, there really is this amazing diversity of shapes, of colors and forms. And I think that really hints at the diversity of lifestyles that these things have. They range from pathogens that kill things to things that are mutualistic or completely beneficial, things that are benign. They take all of these life forms, they run the whole sort of spectrum of life. And the most amazing thing to me about them is that, no matter how different they look, they're all unified by the fact that they're basically entirely comprised of these little strings called hyphae. And so here are two scanning electron micrographs of lichens, and you can see all the little strings of hyphae, the fungal hyphae, that make up the body of all of these fungi, including the lichens. And it just, I think the amazing thing is that these are fundamentally identical. They're just woven together and organized in slightly different ways. So, there's all of these different sort of forms that fungi take and functions that they take. And, one of them is that they enter into mutualistic relationships with other organisms. And mycorrhizae are probably the most well-known example of fungal mutualisms. And so these are the fungi that sort of associate with plants, and the fungus and the plant both get something really beneficial out of the deal. But also, this is basically why we have forests. It's why we have crops. Like the whole world exists basically because we have these mycorrhizal fungi. They're imperceptible, and in some ways, to many of us, they're invisible. But in a lot of ways, they're these networks that kind of link everything together and run the world in like a mycological, botanical internet, right? And so if these fungi are super important because they enter into these mutualistic relationships where both of the partners or all of the partners involved benefit, lichens are another example of that. But rather than linking everything together in this sort of botanical internet, they've evolved this amazing lifestyle that involves the fungus entering into these mutualistic relationships, these symbioses with algae or cyanobacteria for the purpose of obtaining nutrition. And so, the fungus is providing basically the house, and then the alga is living in the house and providing food for the fungus. And these things, what's absolutely amazing about them is that, if you were to culture them out separately, the fungus would look like bread mold or something like that. And the alga would look like green schmutz on a rock or a tree, not very spectacular, right? But yet, when these things get together, which they have to do, at least the fungus has to do to survive. The fungi don't exist free living in nature. They have to enter into these partnerships. They get together and they form these incredibly complex structures. So like here is a section through a lichen, and you can see that there's these layers that are composed of purely fungal tissue. And then there's a layer where the alga and the fungus are intermixed together. That's always the layer that's kind of exposed to the sunlight so that the algae can photosynthesize. And, this might look pretty simple, but it's kind of amazing that these things do this, and then they do it all the time. And then when they get together, they always look the same. So the same fungus associating with whatever algae always looks the same way. And the forms that they take are just otherworldly. There's, the colors they make, the forms they take are unlike anything else in nature. When you look at lichens, especially under a hand lens or with a compound or with a dissecting microscope, it's like peering through into another dimension, or something in outer space, except that it's actually like all around us, which is utterly amazing. And, so lichens are fungi, and they're fungi that enter into these relationships with algae and cyanobacteria. And I think it's really important because this is a question that everyone always asks. Lichens are named based on the fungus. And so every time you see a lichen that looks the same, it's always the same species of fungus that's entering into this relationship. So in this case, this is this beautiful lichen, Anaptychia palmulata. It's an old growth indicator lichen. And the name of the fungus is Anaptychia palmulata. And so when I first started studying lichens, the dogma was that it was one fungus that associated with one alga. So every time you saw this lichen, it always associated, it was this one-to-one relationship between that fungus, the Anaptychia palmulata, and the alga Trabuxia glabra. And it was really only with DNA sequencing, because all these algae that they associate with, don't tell my graduate student who's working on them, but they all pretty much look the same. They're like little green balls that you have to look at under a compound microscope. And even then, they all essentially look very similar, or many of them look very similar. And so, just looking at them, it's really hard to distinguish between them. But with DNA sequencing, they started to sequence the algae that were associated with the lichens, and they found out that it's not always, in fact, it's often not a one-to-one relationship. And so, maybe when this fungus associates or grows in New York, it associates with this alga. But then maybe when it grows in Maine or further north, where it's much more sort of more colder, it might associate with a different alga that's better adapted to that location. And then the same thing, like maybe if it grows in Florida or Louisiana, it'll associate with an alga that's better adapted to those areas. And so, what's really important is it's not the fungus that is driving where these things grow. The fungi appear to be relatively general in terms of their, ability to grow in different places. It's actually their partners that are determining, whether or not they're able to exist in a place, because the algae seem to be more sort of limited in their environmental suitability. And so what we've come to learn is that it's actually, this one species of fungus could associate with different ones depending on where it's growing. But then as we have gotten better and better DNA sequencing technology, it's actually turned out that we're finding that within even a single lichen, there's often more than one alga present and more than one sort of genetic strain or like individual of the same algae present. And, the reason for this is likely that having multiple partners potentially available inside of you that have slightly different tolerances for like temperature and humidity and things like that makes these symbioses better adapted to changing climates now and change in general. So changing environments over really slow, long periods, mean that maybe some alga that's better adapted to a warmer time, maybe it will proliferate in the lichen, and the ones that are less adapted to that will sort of, less in number. But then if it gets cold again, or the lichen encounters some other environmental condition that's better for one of the other algae, maybe those will proliferate more and the other ones will sort of diminish in abundance, but they'll still be there sort of in reserve for, rainy day. Think of it as like a rainy day fund. So, lichens are not just fungi. They're also algae, cyanobacteria, but they're also really a community of all sorts of things. They're like hubs or parties in the forest. Like everything kind of functions around them. So you have your dominant fungus, that's the one that gets the name. You have one or more of these partners that are photosynthesizing inside of them. And then you also have this like whole microbiome of viruses and bacteria and stuff like that. That's the same as ours, right? Like every living thing on Earth has a microbiome, so do lichens. And included in that microbiome are other fungi that are not the main symbiotic lichen fungus. So there's all sorts of other fungi that live in them as well, the same way that we have fungi that live on and around and in us. And there's also little tiny invertebrates like nematode worms and rotifers and tardigrades that sort of live in, on, and around all the little nooks and crannies of the lichen. Again, if you look up close at them with a hand lens or with a dissecting microscope, it's like a tiny mountain range or hill range with all these little folds and nooks and crannies where things can grow. It's like, imagine how diverse the microhabitats are just within a single lichen that might be, the size of a quarter. Certainly not a penny, because we don't have those anymore. And then, in addition to all of these living things, there's the chemicals that all of them secrete and, all of the interactions that they have with each other. And so, it's the lichens really are this amazing community and this hub of different things that are organized more or less around the scaffolding, organized around the building of this main dominant fungus. And so, they often share their partners. And so that's why when you look at all the lichens growing on a branch, like here's a branch in North Carolina where I've done a fair bit of work, and I just labeled some of the lichens on here. These are very different species of fungi that look very different, but it's likely that they are sharing some of their partners amongst the different individuals and amongst the different species there. And so they actually do rely on each other for survival, both for having all of these partners available, but then also because they suck up lots of moisture and release it back into the environment slowly. And so they kind of like a sponge. And so when you have a lot of lichens on the landscape, they create the kind of moist moist conditions that are necessary to sort of propagate more lichens. So like when lichens beget more lichens, that's the way we like to say it. And what I think is really interesting is that this lifestyle that's been so successful, they're everywhere, they're super diverse. We've shot them into space, brought them back to Earth, and they just like seem okay, and they photosynthesize and they're happy. At the same time, they're extremely sensitive to changes in the environment, rapid changes in the environment, to air pollution, to disturbance, to things like large scale forestry and agriculture. And so it's really this paradox that on one hand, they're incredibly adaptable and important to ecosystems, but on the other hand, they're also extremely sensitive, and as a result, many of them are threatened and have declined across their ranges. And so, the work that I do is really focused around two different things. One of them is to study existing museum collections, like those at the State Museum. And the reason for that is that you can't really integrate them into sort of modern understanding of biodiversity and conservation without updating the information. And you can't just sort of change a name, you have to actually restudy the specimen. And an example of that is like there's this genus of crustose lichens called Acrolechia. They look like little donuts or saucers. And so all of the specimens that would have been collected historically would have been named Acrolechia parella, but Acrolechia parella is a European species that doesn't occur in North America. And so you have to look at every single one of them, study them with microscopy, and with that, you'll be able to decide which of the 4 different species that occurs in the Northeast those specimens represent. And of course, each one of those 4 different species has a different ecology, a different conservation status, and potentially a different geographic distribution. And so, what's hidden under one name in a museum collection often actually represents a suite of different species with very different things going on about them. And so in addition to this large scale study of museum collections that I've done throughout my career, I also do a lot of documentation of biodiversity, very similar to what Patty is doing. And a lot of what I do has been to develop and sort of scale up this intensive sampling of 1 hectare plots to develop a way to understand and compare lichen diversity between many different places, whether it's like 2 different forest stands on the same road, a mile apart, or whether it's mountaintops in New York and North Carolina. And so within each one of these plots, which we make sure is standard in its vegetation type, so like a spruce fir forest or, a hemlock stand or something like that, we conduct an inventory where we collect a specimen of every single lichen that we can find in the plot. We also assess the habitat quality using a 1 to 100 score that's based on a number of different components. We took that from forestry and adapted it to our uses. We also DBH all the trees in like a 10 by 10 meter subplot. We quantify how many rocks and how many logs there are, more or less visually. We also inventory all of the woody plants in each of these plots, so we know how many species there are. And so what we get of things that the lichens can grow on. And so what we get is this really nice picture of the lichens that are growing at a site. So, the diversity of lichens, the taxonomic diversity, the number of species. And then we have this really nice suite of information about the site, variables that we can study to try and understand what it is that underlies what is there and where it is. And then we can also use, GIS and other technologies to gather additional variables remotely rather than in the field. And so what, I started this work with colleagues of mine from the University of Colorado in Boulder back in 2016, and we started sampling those plots in North Carolina, and we expanded over a much larger range over the next like 10 years plus. And I'm now working on expanding that in New York as well. And so what we've ended up with is this amazing, unparalleled resource for understanding what is driving lichens, where they are, where they aren't, the species diversity, everything like that. So we have these plots that we've been studying. We have over 1,000 species in those plots, more than 28,000 occurrences of these species, each of which is represented by a museum voucher. We have the specimens that I've studied at the Botanical Garden where I used to work in New York City, and then here at the State Museum and at many other institutions where I've borrowed them from. And that's about 1,400 species in this big area of the Appalachian Mountains and New York. And 27,000 occurrences of those species again. And then all throughout the collections of natural history collections in North America, there's like a total of 1,600 species. Some of those that I've looked at the underlying occurrences, some of them I haven't. And then we also have iNaturalist that my colleagues and I regularly, look at and vet and identify many of the observations of iNaturalist in this region so that we can use those data to also understand what's going on with the lichens. And so just broadly speaking, I wanted to give you a few of the things we've learned from this huge amount of data that we have, right? Like, it's very rare to have hundreds of thousands of records of lichens of over 100 species across a range that extends from Alabama to upstate New York, and sampled in so many different ways. And so one thing that we can do is we can actually look at how the populations and the ranges of these species have changed over time. So here are two rare species, and we can look at their original distribution based on all of the records that we have. We can look at how many of those were remaining after 1934. That's a cutoff that we use for some of our assessments. And then we can also look at the number of those, or where the ones are that are still there. And you can see even just from these maps that there's like a pretty dramatic change. But what's really interesting is we can quantify them. And we can see that actually most species don't change in their overall range size. So like the total area where they are has not changed dramatically over the last 100 or 200 years. And so each one of those bars above a species name corresponds to one of those time points, the original one, 1934, and then present. So most of these haven't changed dramatically in where they are overall. But what's changed dramatically is that for many species, they've declined dramatically within that area. So they've become much, much rarer. And so you can look at that and classify these into whether they're sort of, least concern, lower concern species, or species that are vulnerable, or ones that are critically endangered. And the really, really conspicuous thing that we see across many of these species, especially the ones that are endangered, is that there's the declines that most impact the overall population of the species have been in New York, New England, and the Northeast. As well as some areas of the Southeast. And, the areas in the North are incredibly important because this is likely where most of those species from the South, at least, are going to have to migrate to in the future under future climate change scenarios as the environment is changing. Now, we can tell that there's lots of common species, and we know these to be common based on these data. So species that you all can see on any walk around here: Punctelia verdecta, Flavoparmelia caparata, that's the green one that grows on everything; Lepraria finkii, this beautiful dust lichen; Almondenia polyspora, little black dots on twigs everywhere. These species are really common. We find them almost in every plot. They grow all over. And, again, the data we've got behind it give you a sense of that being the case, right? Like, there's a lot of dots here. The black dots are dots of specimens that I've examined, and the white dots are ones from sort of bigger biodiversity aggregators that are specimens that I haven't examined, but other people have. And because these are so common, it's reasonable to think that most of these are probably accurate. But then we also can tell a lot about rare species. So Dermatocarpon luridum is this really beautiful species that we have that grows around here on rocks and streams. And that's a species that's, not super rare, but it's scattered, and it's rare on the landscape because its, habitat is not super common. Rocks in streams in the right place, and the streams have to be, relatively high water quality. You have species like Rhinodina fryi. That's a species that isn't very common. It's not commonly documented, but that's because it grows on canopy branches of certain species of trees. So you have to collect the canopy branches of the certain species of trees that this little tiny lichen grows on. And then you have things like Basidia schweinitzii, Pyrenula pseudobuffonia. These are two crustose lichens that are more or less old growth dependent or mature forest dependent. And so these are species, again, that are relatively widespread, but they're much rarer than the common species that I just mentioned. And so, using all of the plots that we've looked at from Alabama through New York, what we can see is that each one of these white dots is a plot that we've studied. So each one of those is 1 hectare where we've gone and spent hours, like collecting everything, studying it, getting all these data. And what we can take away from it is many things. But to me, the most striking thing is that we've learned that diversity of lichens is really unevenly distributed, and it is highly skewed in where it is. And, the hot colors are high diversity and the not so hot colors are low diversity. And what's really striking about this is that there's, markedly lower diversity in Pennsylvania and parts of New York. And so the question is, what exactly is the underlying reason for that? That's something we're working on now. Something that we know to be the case is that lichen diversity is strongly driven by habitat quality. This is work that my colleague Laura Bacchus has done. It was published in Biological Conservation. And basically, no matter how you look at it, no matter how you split up the species, habitat quality is the strongest driver of lichen diversity at this point. And habitat quality means a lot of different things, but think of it as though, a planted forest, planted pine plantation is going to be a relatively low habitat quality relative to a mature old growth forest. And so what you can see here is a graph of number of species in a site versus habitat quality. The interesting thing is, though, that the habitat quality, if you look at it around 60 to 100, again, I can explain that score in more detail, but you don't really need to know the details of it. Just imagine that 60 would be a decent forest that you would want to take a hike through, and then 100 would be like the ideal old growth forest that we would all love to spend our time in. And so once you get to that sort of like having a healthy, decent forest, you basically end up with, habitat quality no longer being the driver of the lichen diversity there. And instead, what we find is that what drives patterns of diversity and occurrence of species are two primary things. One of them is the partners themselves that are inside the lichens. So there's like 3 main kinds. There's a lot of ways to understand this, but there's 3 main kinds of partners: the cyanobacteria, Trentopolia, which is a kind of green alga, and then other green algae. And basically, these are graphs showing the diversity of lichen species with those different partners by elevation for all of our plots. And you can see that they have very different curves, right? Which suggests that, cyanobacterial lichens are less diverse at higher elevations. That correlates to colder temperatures. Trentopolia, they're really diverse at high humidity and lower elevations. And then these other green algae, they kind of have a diversity peak in middle elevations. So this just gives you a hint that really these internal partners are what's driving a lot of whether or not these things grow in a given place. The other thing that's really cool, though, that's completely unexpected that's come out of this work, which was led by my colleague Christine McCain in Colorado, is that we took all these data we've got so many different variables. And, when we put it all in models, what we find out is that the biggest factors of lichen diversity, other than habitat quality, once you control for that, is actually the diversity of native woody trees and shrubs, combined with something to do with the environment, like temperature or precipitation. And so temperature, precipitation, those environmental factors are really well understood to drive diversity of species in general. But it's this additional thing, the additional factor of living things, the diversity of all of these different tree species and shrub species, that's really key to having lichens and having a lot of them. And that's because the bark of different trees and different shrubs is really different, right, from one species to another. And lichens are microhabitat specialists. So different species grow on different bark types. And that kind of intersects with the habitat quality thing, because forests that have higher habitat quality are usually more mature. That's part of the metric. So they tend to be more mature. And mature forests have a broader range of different age classes of trees. And, a sapling has really different bark than a mature tree, right? And so imagine having each one of those different age classes with its different bark types present in the forest, and then also many different tree species present in the forest. And that just gives you a lot of different places where the lichens can grow. And so what I kind of wanted to end on was just that, there's a lot to go into here, and there's so many things that we could talk about. But I just wanted to highlight that there's major past declines that have clearly also happened for bryophytes and other things. And that the declines to these lichens almost certainly have resulted in major unseen changes to our ecosystems. Like, imagine a forest with lichens and a forest without lichens. There's clearly an impact there, right? They're doing all sorts of important things. Also, habitat quality drives everything about lichens, and there's apparently a tipping point, and that tipping point is a really important management goal if we're going to think about, sort of what are the ideal conditions we want to, aim for in managing and conserving our land. And then also partners are really important, and they need to be considered, and they're really key to the conservation of lichens, and so is habitat quality. And then also native woody tree species are really important, and as well as having a diversity of species and ages. And that's really important because it links to all of these different past changes that we've, had on the landscape, but also how these ecosystems are changing with the spread of invasive species, right? And also the declines of many native woody species through things like invasive pathogens and pests. Think of ashes and hemlocks, for instance. And then also just, surveys in New York and New England are really desperately needed. Most of the work that I have done in the past has been in the Southern Appalachians, Central Appalachians, the coastal plain of the Southeast. And it's really only in the last few years that I've moved here and started to get this up and running in New York as well. And I can tell you that the analyses of the sites that we have sampled here show basically the same things are driving lichen diversity here. The same patterns are true elsewhere in the Eastern US. It's just that the diversity here seems to be lower. And the question is exactly why that is. And to do that, to understand that, we have to really document our diversity and do more surveys. And with that, thank you all. Hopefully there's a time for some questions.
Thank you. We're at time. If you want to stick for a minute or two, you guys did a great job of addressing the questions that were in the chat. Great multitasking. So thank you. There's a question about a lichen field guide. If you have a recommendation.
Yep, I'll put that in the chat. Yep.
Awesome. Thank you.
Sorry, I wasn't monitoring the chat at the same time as.
Well, you answered some questions that were right there in the chat. So I thought you were.
I think that was me doing that. So
oh, thank you.
Yeah, no problem.
Well, yeah, I saw you answer, Patty. Thank you.
Oh, okay.
That was great.
Yeah.
There was a question about the habitat quality. What can be done to improve the habitat quality of lichens?
So what can be done is that preserve our forests. Like, honestly, it's a matter of conserving the forest and also having large blocks of contiguous forest. And, look, that's something that's, maybe it sounds silly because I feel like that's how so much of what we do with land conservation now is focused on is, corridors for migration, thinking about the big picture rather than just protecting a little postage stamp surrounded by, development or agriculture or something like that. Like, we want intact natural landscapes. And so that's really what we need to be aiming for with the lichens is that they need these large areas of intact forests, and they need a mosaic of remnant older forests, whether it's true old growth or whether it's, sort of the back 40 of the farm that wasn't cut down, right? But now is surrounded by regenerated agricultural lands in a lot of places. And the other thing is air pollution is really the other big thing. So you can have an old growth forest, but if it's near a source of air pollution or it's been subjected to air pollution, it will have virtually no lichens in it. How do they grow on their rocky and woody hosts? They just attach to them. They hold on for their dear lives. That's not a joke. That's really what they do because they get all their food from their internal food source. So, they're just there to hang on and be the clothes of the forest, which is what they call lichens in parts of Asia. Sweaters for the trees.
Well, that's a great note to end on, conservation. Thank you so much, Patty and James. This was really great. Thanks for sharing your experience and your expertise and your beautiful pictures, absolutely beautiful pictures.
Oh, oops.
And thank you all for being here with us this evening. Tomorrow is the start of spring, and so this concludes our winter speaker series. Hope everyone has a healthy and safe spring and summer and fall, and that you can get out onto the land. And we will see you for our winter speaker series next winter. Thanks again, Patty and James. This was great.
Thanks
Take
for
care.
having us. It was really fun.
Good night.
Yeah.
Yeah. Have a good night.